Okay, hello everyone, greetings. So, welcome to this week's session of this algorithmic thinking in bioinformatics course into some real action today, and then start looking at a very simple problem. That is why this is called an algorithmic warm up of finding frequent substrings in a string, and how do you use that to answer a intriguing biomedical question or a biological question about how DNA copies itself. Okay. So, where in the genome does DNA replication begin? So, that is a question we would answer, and then a simple string algorithm is what we would use to illustrate this. And so, like I said, today is a interesting question about the origin story of DNA replication. So, how DNA replication knows where to start in the genome, and the associated computational problem. We will iteratively formulate the problem starting with this question, and how we can use computational thinking to solve this particular problem, and then its simple algorithmic solution. Recall what we had seen in the last session about central dogma of molecular biology, where we made several simplifications and simply said this two-step process, right? This DNA makes mRNA or RNA, which is this process of transcription, and then RNA makes protein, the process of translation, right. So, that is the simplistic view we provided, and we said that there are many exceptions to this simple two-step view, and then one such exception, which is actually an important exception, is DNA making DNA, right. So, that is the idea of replication. So, you can put it like this, and then this you call it as DNA replication. This is not an exception, by the way, right. So, it is just the way that we presented, we did not point out last time, but it is one of the central processes within a cell. So, whenever a parental cell divides into two offsprings, two daughter cells, the DNA had to be replicated. It has to be made an exact copy, whatever in the parental DNA, the genome has to be made exact copies, and the two of them are then split into the and shared among the daughter cells, right. And in many cases, I mean, barring certain mutations that happen in certain diseases, in many cases you have the DNA, identical copy of the DNA is being made. And if you just think about it, it's really an amazing feat that the cell does, because if you have a human cell, we have 3 billion letters, and all those 3 billion letters, even just copying it takes a few hours of time or a couple of hours of time, and it has to make an exact copy of all those 3 billion letters without any changes in even one of those bases, right. And so, oftentimes you do get identical copies, and that is how like a single embryo then divided and formed all the cells within our body, right. So, a key step in cell division or cell cycle in essentially in development is basically DNA replication, and the question is, where does it begin, right. So, there are many molecular machines that do DNA replication. Yeah, so by molecular machines I mean these different multi-protein or single protein units. So, you have helicases, which is shown in slight orange color, and then DNA polymerases, which is shown in blue color. So, helicases separate out the two strands, and then the DNA polymerase then makes a copy of each of these strands, right. And so, there are these different molecular machines which help in the process of DNA replication. But then a key question is, how do these molecular machines know where to begin, where to start this process, right. And that is what we will focus on in this session today. We will look at how to approach this biological question using computational thinking, right. So, and I would like to comment that most of the subsequent slides are from these instructor resources that comes with the textbook for our course, or the reference book for our course, Bioinformatics Algorithms and Active Learning Approach, by Philip Compo and Pavel Pevsner. So, we are grateful to both of them for agreeing to or allowing us to use these slides in this particular course. So, let us clarify and simplify a question first. So, one is before we get into the question of where it begins, let us just first understand how complementary base pairing helps in replication of DNA. So, we have seen again in the glossary that A always pairs with a T and G always pairs with a C. So, this kind of complementarity of different bases, on which basis it pairs with, how does it help in DNA replication using a cartoon picture, and then where does it begin in a small circular genome. We will not deal with the complexity of human genomes, where there may be many different replication origins, right. So, we are going to look at a very small circular genome, such as that of a bacterial genome, and then see how do we find where in that particular genome a replication starts. Okay. And even the simpler question has key applications in applications such as gene therapy. Okay. So, even the simple question, if you are able to answer, then that will already be useful in of itself. Yeah. So, here is a little cartoon of how the complementarity of the bases, right, how the complementary base pairing helps in replication. So, this was even commented by Watson and Crick when they released their 1950s paper, saying that okay, we have this two strands which are in this helical structure, and it does not escape their attention that this could be used as a mechanism for replicating the DNA. So, how so? You have these two strands and then they are connected by these weak hydrogen bonds, right. So, you just separate out into these individual strands, and then you have these molecular machines which will use these free nitrogenous bases, and then basically use this pairing mechanism that A always pairs with a T and G always pairs with a C, and similarly on the other side to make an exact copy of two more strands, right. So, after you separate out, then you can independently work on each of these strands and then attach these complementary bases, so that you start with one of these double stranded DNA and then you end up with these two of these double stranded DNA, right. So, that is how complementary base pairing works, and that is how the genome is replicated for a cell to divide. The question of interest today though is, where does it begin? And then we know that based on existing biological knowledge that it starts in a region, in a particular region, it does not randomly start anywhere in the genome, right. So, for small circular genomes there are particular regions in the genome where it starts, and then this is called the replication origin. We will represent it as ORI, we will abbreviate it as ORI in the subsequent slides. And you can see here that this is the origin of replication, and then it slowly in a circular genome, it slowly these two strands are separated out, and then the copy, this new DNA which is shown as a red fiber is made, right. So, the red strand is made in both the top and the bottom separated out strands, and that is how the overall you will get in the end of this process, you will get two circular DNAs, right, two circular genomes. And we can think about it now on how can we find out where it begins, both experimentally and computationally. Okay. So, and that is what we will first look at. So, what is the hidden message in the replication origin, and can we use some of these hidden messages to figure out where the replication origin is in this in this genome. Okay. So, first we will focus on something smaller, that we will look at a region where we will assume that somewhere there there is a replication origin, right, and then we will look at whether there are any hidden messages within this replication origin, and then we will backtrack and see if we can actually find those replication origins even if we are not given any ballpark hint on where it might be in the genome. Okay. So, let us first assume that we roughly know that some 100 nucleotides or 100 bases is where this replication origin is located, and let us just understand whether there are any patterns within that known replication origin, and later on we can find the replication origin in the first place. Okay. So, like I said, when you are trying to think computationally, we need to be concrete. So, let us just try to formulate a concrete problem. So, finding ORI problem is finding this origin of replication in a genome, input is a genome and the output is a location of ORI in the genome. That is what we are interested in finding. So, in that case, actually a simpler experiment would be that you just cut out different DNA fragments from this genome and then see if it replicates, right. So, even without knowing what the particular sequence is or pattern is, you will be able to say that okay, I cut out each, let us say each 10 bases in the genome, and then like which 10 bases I cut out and it does not replicate, that can point to some hints about where the replication origin would be. But it is a very time consuming experiment, as also the other probe experiment. Even in circular you can try that argument. That is right, yeah. So, we will have to try all possible sliding windows, whether it is linear or circular, it is roughly the same, I mean it is the same complexity, linear complexity in terms of the number of experiments you would have to try. It will be the number of positions in the genome, and so that many number of experiments is time consuming. And so, can we actually use computation to prioritize which location a motif would appear, right, or which locations, can we prioritize which locations an origin of replication would occur. Okay. So, but then before that we need to frame it as a computational problem. So, this as stated is not a computational problem. We do not know, like a computer scientist who is new to this area would not know what a genome is, and they would not know what an ORI is. So this is not a computational problem. And so to try to frame it as a computational problem, let us look at it a little bit more into actually one known replication origin in a bacterial genome that causes cholera, called Vibrio cholerae. And so based on previous studies, we know that this 500 nucleotides is the region which contains or which represents the origin of replication for this particular bacteria. Okay. And let us see if this contains any patterns or motifs, so that we can use that to prioritize which regions to look for in the whole genome of any new bacterial genome that comes up. Okay. So for this particular bacteria which causes cholera, we know that this is the replication origin. This part of 500 nucleotides of the genome is the replication origin or ORI. Okay. And the idea is that there must be some, the basic premise of our method would be is that there must be some hidden message within this that will tell the molecular machines to actually start replication here, right. So the molecular machines are proteins themselves, so they have to bind, they bind to some of these regions, and then they start the process of replication, right. So there must be some hidden message in here. So these molecular machines, again based on previous studies, we are saying that they do not bind randomly to any kind of DNA sequence. They have some preferences of certain sequences that they like to bind. And so can we then find that hidden sequence or a hidden message by just looking at this text without knowing a priori what that pattern is, right. If we know that pattern, that okay, it binds to AACC, then we can scan through this and look for AACC, and then we will be done, right. But if we do not know a priori what is the sequence that these proteins or these molecular machines like to bind to start DNA replication, then how do we solve this problem? Okay. And so this is how we have. So far based on our understanding, so we are going to find a hidden message in a string text representing the replication origin. Okay. So this is a sub problem, a subroutine if you will. The original problem is finding replication origin, but this hidden message problem is given an origin of replication, right, given a string, are there any hidden messages in there? Okay. So this is a subroutine if you will, a sub problem. So finding a hidden message in a string and a string text which represents the replication origin. So this we already know for certain organisms like this cholera bacteria, and then output is a hidden message in text. And like shown here, this is again not a concrete computational problem, because what do you mean by hidden message? It is very ill defined, right. I mean it is not defined precisely. Text is fine, you have a string text of ACGT letters, that is fine, but what is hidden message and how do you find it, right. And it's more like this Goldberg problem. So there is this a secret message that left by pirates, right, and then a certain work by Edgar Allan Poe, where the pirates have this message and then they are not given any concrete problem on okay how to decode the message to figure out where the treasure is, right. And one particular clue they found out was, if you just stare at this long enough, you will see that certain 3 letters that keep repeating. Can you see any 3 letters that consequently keep repeating? The 3 letters which are together and that keeps repeating, we will call it a tremer that keeps repeating. Is semicolon 48 repeating often, more often than the others? Okay, I will show it to you. So you can see here that semicolon 48 repeats a lot more than other 3 letters. So it may stand out if you stare at it long enough, and this is, we also know an additional information that this is English language. Okay, and so the most common 3-letter word in English language is the, and so you can then say that maybe semicolon stands for T, 4 stands for H, right, and 8 is indicating E. Using that you can replace everything with THE and then try to decode the whole message. Okay. So but you can see that this is a lot of guesswork, and it's not very concrete or well defined, right. And so basically we need to somehow make it more precise, and we are going to get this hint from this hidden message that we just saw, that there are these frequent words, right, and frequent words also, or frequent subwords of a long string, right. So frequent subwords within a long string also make sense in biology, because if you have a particular molecular machine again. So, in bacteria these are called DNA. So, these are proteins that find the replication origin and then starts the replication process by melting the DNA, right. So, these molecular machines basically need to bind to the region where the replication needs to start, and then if you have many more patterns to which they bind, there are certain substrings to which they bind, and if there are many frequent occurrences of this substring, then there is more likelihood that the protein will bind to that region, right. So, the substrings to which these molecular machines bind, you want those patterns or also known as motifs to occur frequently within this replication origin that is provided to us, and that we can hypothesize to be the hidden message. Okay. And so, for instance, here in this particular string, this is let us say the replication origin, you can see that AATTT is surprisingly frequent, right. So, if this were a random string, seeing 3 occurrences of AATTT will be really unlikely, right. So, in this you see these 3 occurrences. So, this is really frequent in this particular string, right. This particular pattern or motif AATTT is frequent in this particular, frequent meaning it occurs 3 times, right, in this particular string. And so, can we use this frequency of these substrings to actually make our problem more concrete, and this also we will along the way see if it makes biological sense. For now it makes biological sense because these molecular machines have to find many of these patterns to reliably bind to a region to start replication. So, the way that we have rephrased the problem now, the hidden message problem has now become the frequent words problem, right. Finding the most frequent k-mers in a string. As I said, k-mers are simply substrings of a string of length k letters. Okay. So, a string text and an integer k is provided. So, we have some prior knowledge on how long of a pattern we are looking for, right. A string text and an integer k, all most frequent k-mers in text. Okay. So, that is what the problem stands now, and this seems good. I mean, in the sense that it is more computationally understandable. A computer can, I mean a computer scientist can understand it and can code an algorithm for it and try to apply it on an origin a genome sequence and try to figure out what is the most frequent words, right. But still we need to clarify what do you mean by all most frequent k-mers, right. So, we need to be precise there, and a frequent k-mer or the most frequent k-mers is a k-mer that is more frequent or as frequent as any other k-mers in the text. Okay. There is no other k-mer that is more frequent than this particular k-mer. So, that is what is written here. So, a k-mer pattern is most frequent k-mer in a text if no other k-mer is more frequent than pattern. Okay. So, there is no other thing which beats this. There may be ties, there may be 2 k-mers, 2 distinct k-mers which occur the same number of times. Okay. Like I said, there are certain molecular machines which perform DNA replication. In eukaryotes, we have seen polymerases, helicases and so on. In bacterial genomes, it is a protein called DNA. Okay. This is a replication initiation factor as well as it starts, it begins the replication process. Okay. And once it starts it, then DNA polymerase and other proteins or enzymes can come in and proceed with the process. Okay. And we know from earlier studies that typically like very short nucleotide long words are bound by this DNA. These are called DNA boxes. DNA box is nothing but a substring of length roughly 9 that this DNA protein binds to and then starts the initiation process. Okay, starts the replication process. And the DNA box is a hidden message basically telling DNA that bind here, right. So, in very simple terms, the DNA protein doesn't, or in other words does not randomly bind to any 9-mer sequence. It has certain 9-mers that it likes to bind, prefers to bind, and those are the ones called DNA boxes. Okay. And for it to reliably bind, to not miss any things, we need to see multiple DNA boxes so that it can bind to one of them, right. So, that is where the frequent occurrence of DNA boxes is the biological motivation, and then that is how we have converted this into the computational problem. So, now how do we solve this? I do not have to walk through the steps of the pseudocode, I mean the algorithm, or provide you the full pseudocode. You have already taken programming and data science kind of multiple courses in this program, right. And you may be like, if you are asked to come up with an algorithm for given a text and an integer k, all most frequent k merged in text. So, with someone with a programming data structures background, you may be able to come up with solutions that are one of these 4, okay. And they are in increasing, these are the run times of these different algorithms you may have thought about, right. So, and each one of them is in this big O notation, right, and that also again you may have seen in the earlier algorithms classes. So, it just is an asymptotic running time of these algorithms. So, some of the algorithms, some of you may have thought about an algorithm in big O of text squared times k, some of you may have thought about big O of 4 to the k plus text times k, and so on, right. The fastest is just linear time, right, it just takes time, that is the number of letters in text is the order of that, is the running time of the algorithm, right, big O of text. So, this is the number of letters in text is what is written here, right, cardinality of text. And so, what we will do in the next 2 slides is just look at these first 2 algorithms, right. So, I hope you can already see what the algorithm may be, you can probably think about it before you go to the next slide, right. And then we will leave the others as exercises for you to think about. Okay, how do you get something like text squared k, right? One approach you could think about is the following. So, you have a string of length n, which is size of text. So, this is the string text, right, and then you have all these k mers that you can do in a sliding window, each one of them is k letters, right, you can keep on doing it. The last one will be, so if this is the first k-mer, this is the second k-mer, third k-mer, and so on, and then the final one will be the n minus k plus 1th k-mer, right. At the most all of them can be distinct, right, but we would expect that many of the k-mers will keep occurring multiple times in this whole string, right. And so, what you can do is you can allocate an accounts array, right, that will have as many arrays. So, this is a counts array that will have as many entries as there are the number of distinct k-mers, right. And for each one of them we are basically going to scan through the rest, right. So, for the first k-mer you have a particular letter, let us say that k is 5 and then you have AATTT, right. We are going to scan through the whole text, right, and then write down what is the number of times that AATTT occurs, okay, and that scanning you write a loop around and you scan it, and that takes order of n time, right. So, for this to fill out what is the count for the first k-mer, first k-mer's count, we have order of n time, right. Similarly, for the second k-mer's time count you have order of n time, and so on, right. And in order to figure out if AATTT, this is length k, right, I have intentionally made an error here, in the sense that for each of these counts, right, I am saying that this can be figured out in order of n time, because you have to take this AATTT and you have to slide it through the whole text and figure out how many times it occurs, okay. This sliding is going to be order of n times, so that is order of n, but then each of the comparison, let us say I want to check if this is AATTT, I want to know whether the letters in the text match AATTT, then I have to do k comparisons, right. So, I have to do k operations. So, this is actually not order of n, this is actually order of nk, right. Similarly, for the next one maybe it is AATTTG, then for the second k-mer again I have to run through all of the locations in the text, and then that would be again order of nk, right, order of n locations, at each location I am doing k comparison. So, it will be order of nk. Similarly, there are order of n or n minus k plus 1, I can simplify it as order of n possible distinct k-mers, worst case, right. So, then my total running time is all the possible k-mers times order of nk, right. So, that is order of n squared k, which is what is written as order of text squared into k, right. That is what was in the previous one. So, that is the first algorithm, which is simply scanning every location schemer through the rest of the string, and like I said, we are just doing a very simple algorithmic warm up. So, we will look at other algorithms later on, but this is just to make sure that all the notations and big O notations. So, we will actually call this as big O of text square times k, right. So, the asymptotic running time of the algorithm. Let us now look at how do we get this. I mean, this looks like a quadratic in the length of the text. If the text is really large genomes, this looks painfully slow for it to run, right. Can we do something that is linear in text, like not text squared, but just text? So, then that is this, if k is small, assuming that k is small, okay. So, that k is 5 or k is 4 and so on. So, then that is the algorithm that we are going to do, that will be big O of 4 to the k plus text times k, and can someone say what that might be? We are aiming for 4 to the k plus, that is right, yeah, that is precisely the algorithm, thanks. So, you again have a counts array, but then now the counts array is 4 to the k in length, it is not text in length, right. You have the first one is going to be all A's, right, second one is going to be all A's, and then the final letter is going to be changed to C, right. So, maybe I should write it down here. So, first is going to be, let us say A, second is going to be A, C, A, G, A, T, and then you kind of make it A, C, A, and so on, right. So, you just write down all these 4 to the k k-mers, right, and then you have a hash map. So, from here you will enter into this location, the first location, and then from here you will enter into the second location and so on, right. So, each of these 4 to the k will have a hash map into an array of length 4 to the k, and that will be the counts array. Now, you initialize all of them to 0, because that is if you have not seen any of the text, all of them are initialized to 0. Then you have your text 1 to n, and then you look at the first k letters in the text, you apply this hashing, right. Again, this is a data structure you would have seen earlier as part of this program. So, you apply a hashing, you get a key, and then that key will correspond to the first location if it is all A's, right, or whatever letter is here. If this is A, T, 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 then it will correspond to some specific location in this count, and then you increment that count, and then you keep on going to the next location, the third location, fourth location, and so on, all the way till the n minus k plus 1th location, right. And then each operation to compute the hash, you have to read all the k letters. So, order of k per location in the text. So, you have order of n k, right, for computing the hash, and then to set up and initialize this array and to figure out what is the largest elements, the max count of this array, then that is 4 to the k time, right. After all of this has been scanned, you have to go through this and then figure out what is the maximum element and report that, right. So, that is order of n k plus 4 to the k, that is what we are looking for, order of 4 to the k plus text times k, okay. So, this is much better than text squared k if you have larger genomes and k is small, okay. So, that is the second one, and like I said, we just looked at this one before we looked at this one, and these things can also be done. I will leave it as an exercise, or we will actually look at this text one later on in the course. So, there is some interesting data structure called suffix arrays and suffix trees that will help you solve this in linear time, and this one I leave it as an exercise, and even this there are also simpler approaches. So, if you have seen string algorithms before, you may be interested in trying this out, okay. So, that is where we are at in terms of we translated this problem of finding replication origins in a genome to look at the sub problem of given a replication origin sequence, what are some hidden messages, and we post it as a frequent k-mers in a string problem, right. And now we need to figure out, okay, how frequent is sufficient, right. So, how frequent is frequent, right. So, are they really surprising for a 500 long string is occurrence of 3 times more frequent or 6 times more frequent. So, let us try to give some intuition around it, okay. So, here is our algorithm applied on this ORI of Vibrio array, right, this 500 nucleotide or origin of replication, and we got 3 as the maximum frequency, right, and then all of these 9 mers are appearing 3 times, right. So, now we are left to wonder what is the real hidden message, or what is the real box, DNA box that the DNA binds to, right, this molecular machine DNA binds to. And so what we can do instead is try to understand this reverse complementarity of the DNA, right. So, you have both a forward strand and a reverse strand, and so you can look for, what I have shown here is only one of the strands, right. So, you can also look for messages on the other strand, and so each one of these 9 mers will also have a reverse complement, and then we can also then club both of them together and count it as for one k-mer, right. And then we can ask the question, is it statistically surprising to find a 9-mer including this reverse complementarity in a string of length 500 nucleotides, right. So, this 9-mers, all of these are appearing 3 times, right. In a random string, is it expected that these things will appear also 3 times or more, right. And for that you can look at again this n, right. So, 1 to n is 500, right, and the number of k-mers that appear here are n minus k plus 1, and roughly it is 500, let us say, and k is 9. Going to just approximate it as, instead of saying 500 minus 9 plus 1, I am just going to say it is 500, right. And then if there is no hidden message here at all, right, you can assume that every position is going to be A, C, G or a T with a uniform probability 1 over 4 each, right. And again there are probability foundations that you have done earlier in the program. So, I am going to assume a basic understanding of probability going forward. So, each one of them is going to be, each of these location, if it is a random string, is going to be A, C, G or T with probability 1 over 4 each, right. And so, for something like AT, GAT, CA, AG, right. So, for all of this to miraculously happen, even if there is no true hidden message, but for them to just spuriously match a random string, is going to be, each position is going to be, this first position is going to be A with 1 over 4 probability, second is going to be T with 1 over 4 probability, and so on, right. So, and so it will be 1 over 4 multiplied to itself k times. So, the probability that at any one location, probability that at location I, let us say, location I matches a particular given k-mer, matches a given k-mer, is going to be 1 over 4 to the k, right. And so, on average, if you multiply across all of these, so the average matches of a k-mer in a n long string, right, n length string is going to be n over 4 to the k, or n minus k plus 1 over 4 to the k to be precise, right. That is the number of k-mers that can occur in this string, which is approximately n over 4 to the k. And so, in this particular case, it is going to be, if you plug in 509, right. So, this is the, on average, that the number of frequencies of any particular k-mer that you will expect in a 500 long string, right. And so, this you can see is a very small number, and so given that, it is indeed surprising that a 9-mer appears 3 or more times within approximately 500 nucleotides. Okay. So, all of these are statistically surprising, but can we still zero in on one of them, right. And to do that, we are going to use this reverse complementarity, right. So, again due to this complementary base pairing, so AT, GAT, so A pairs with T, G with C and so on, right. So, this forward strand k-mer is equivalent to this reverse complementary k-mer, right. And so, we can then combine them both into one unit and then try to get the frequency of that unit alone, right. So, and essentially the biological motivation is that the DNA, the protein does not care whether it is in one strand or the other strand. So, whenever it finds this motif, it binds. Okay. So, we can then group these two together and then count them as one unit. If you do that, then you will indeed see that two of them are actually reverse complements of each other, right. So, if you see this, this CTT, GAT, CAT is actually the reverse complement of this first string. So, this occurs 3 times, this occurs another 3 times. So, 3 plus 3, this unit occurs 6 times. Okay. So, so basically you have something of this k-mer occurring 6 times, and that is increases the statistical surprise, right. And so you have this as the likely candidate for your DNA box, the region or the substring within this string that DNA protein binds to start the replication. Okay. Okay. So, now can we just switch from cholera bacteria to some other bacteria, Thermatoga petrophila, right, where we do not know much, let us say. So, some new bacteria that comes in from a hot spring, let us say, right, and then we want to know hidden messages. Can we apply the same algorithm here, right? Can we just take these sequences and then say, okay, these are the DNA boxes and apply it here, right? But the problem is the same messages may not, the same motifs may not apply for all the organisms. So, we have to apply the frequent subwords or the frequent words problem again here, right, and then no single occurrence of these two motifs that we found here in previously in the cholera bacteria occur here, right, and that is not surprising to a biologist. It may be surprising to a computer scientist. Biology is all complicated. So, the machinery may not use the same pattern or motif in different organisms, right. And so, we cannot basically scan for these known motifs from other organisms. We just have to apply the frequent words problem again to this particular replication origin, right, and then we get these different numbers of words, right. So, all of these k-mers are all appearing to be frequent in this particular sequence that is given here, okay. And this is just reiterating that different genomes will have different DNA boxes or different hidden messages. And you can build on this simple problem. So, we just for instructional purposes we are just keeping it simple in terms of frequent words is what we are looking for, but you can make it more complicated. You can look at these reverse complementarity pairings and you can look for other messages and then try to get at the real signal and use some other features such as the distribution of GC and AT in these different sequences. They call it skew. So, those kinds of properties can be used to focus on in the actual candidate among all of these candidates, right. So, one idea that we said was this reverse complementarity grouping, and then this particular one is what there is a software that implements these different heuristics for finding replication origins called the OriFinder software, and this is the one that it finds. Okay, this is a candidate hidden message in this particular Thermatoga petrophila. Okay, you can see that there is 1, 2, 3, 4, 5 times it occurs in this particular sequence. And this also kind of illustrates why a computer is needed, right. So, just looking at it, it is very hard for a human brain to recognize patterns here and find frequent k-mers in this particular string. So, but very simple to write a very naive algorithm to figure out what the most frequent k-mers are. And then once you highlight it, yes, our brains can see, but you can see where the kind of the routine repetitive works that if we properly algorithmically code it, we can find these messages. Okay. So, we have so far only solved the sub problem, right. So, we have only solved the sub problem of given a replication origin sequence, what are the hidden messages within it, what are the frequently occurring words. Okay. But of course, if we start with a new species, new organism, we do not know where the replication origin is in the first place. So, how can we bootstrap from this procedure and try to figure out where the origin of replication is? Okay. Especially the key word is a long genome. If it is a short genome, then we can basically look at different places where the origin of replication may be, and then try to hypothesize one of them is the origin of replication and try to see if it contains statistically surprising number or count of frequent words. Okay. So, this is where we enter into this figuring out clumps of hidden messages. Okay. And let's see what it is. The strategy is as before. Okay. So, we are going to apply the sub problem in a box. We are going to assume that this is your whole long genome, right, and then we are going to guess different boxes where the origin of replication may be. So, this is one guess you have made. Then within that we are going to run our sub problem to figure out if there are any hidden messages, any frequent words, right. So, and then we are going to just repeat it, right. So, here we found like one particular k-mer that occurs 4 times. Okay. So, then we are going to repeat it if we know it, that's great, but then if we know don't know it, we are going to guess these different boxes where, I mean, different boxes replicate these different potential ORI sites. So, this may be one potential location of an origin of replication, this may be another potential location, and so on. We are going to try different origins of replication, and within each one of them we are going to run our frequent K-merge problem, right. And then we see here that this K-merge is only occurring one time, so it is not statistically surprising, so you do not look at it. This one is occurring 2 times, again in a certain long, like a 500 nucleotide long or whatever that length of the box is, right. So, we do not find it statistically surprising. Here it is 4, so maybe there is something there. Here it is 5, one K-mer appears, the most frequent K-mer appears 5 times in the string, right, and here again it is 5 and here it is 4, right. So, now you can look into the boxes where there are these highest number of these frequencies that you are seeing, and then see how they are clumping. Okay. Maybe this region is where the actual origin of replication is, because there are 2 of the messages, hidden messages in 2 of the boxes, and both of them are clumping together. Okay. So, that is what the next problem is. A K-mer forms a clump inside a genome if there is a short interval of genome in which it appears many times, right. So, that is what you mean by a clump. Its occurrence of the K-mer is very spatially closer to each other. Okay. But of course, this is not computationally concrete yet, we need to make it concrete. One way we can make it concrete is a K-mer forms an L comma T clump, right. You have 2 parameters, again we will have to know it from prior biology on what values of L to use, what values of T to use, but you have some parameters L and T. So, you have this L comma T clump inside a genome if there is a short length L interval of the genome in which the K-mer appears T times, right, T times or more. Okay. That is what this L comma T is, and then again you can write it down as a problem and you can think about algorithmic ways to solve this. So, you can write a pseudo code, again these are very simple problems. So, you can after a few, I mean after some amount of thought you should be able to write a code to answer this problem, right, to solve this problem. So, you have a genome, a string genome, and then you have a string genome and an integer k, length of a pattern, L is the window length, right, and T is the number of patterns. So, the 3 parameters k, L and T, you want to figure out all K-mers forming L comma T clumps in the genome, right. And the challenge is of course that with the simplistic problem formulation, so there exist more than 1000 different 9-mers forming 500,3 clumps in E. coli genome, right. And so this does not really narrow down the possibilities of which one is the true one, and so it will be harder then even to prioritize which ones to follow up on an experimental, let us say you want to cut that region and see if it replicates or not, right. And basically there are certain other strategies that this REC finder software uses to prioritize these thousands of regions. One of them is this Q that I talked about. So, the frequency of GC nucleotides compared to the frequency of AT nucleotides in a sequence, and that Q is present in certain regions, has a certain pattern of increasing and decreasing near origins of replication. So, you can use that to figure that out, but we will be getting into too much biology there. And but there are some useful insights that you can gain from understanding the biology, and then using that, and REC finder software and other kind of efforts use some of those signals. Okay. We are not going to delve into it. I am just going to end with a final word about frequent words with mismatches. Okay. Oftentimes these molecular machines are not very precise, right. So, I mean it is not like the DNA will say that I will only bind to AATTT, right. So, there is a bit of a buffer, a tolerance in which it may also be able to bind to AATTC if that is this last base is not a very critical base for the binding affinity to happen, right. So, we should also be willing to allow mismatches when we are counting the most frequent K-mers, and that is what the final part of this is about, algorithmic warm up is about. So, we are searching for a bit more elusive hidden messages, elusive in the sense that they are not exactly occurring, but there are some errors which may happen or some buffers or tolerance levels we may want to bear with, right. So, we may want to allow. So, we have seen earlier that this origin of replication 500 nucleotides in this cholera bacteria has 6 of these DNA boxes, 3 of them in the reverse complements, right. Can you find more of them, right. And then if you allow these mismatches, like a single mutation, let us say you allow one of the letters to change. So, here for instance the final letter is changed here C, right, So, this is ATGAT CAAG, that final letter G is changed to a C, and similarly the second letter is changed to a A. So, these are mutations, right, single point mutations. And now if you look at this, then these are more number of occurrences, right? There is 2 more occurrences here. These are exact occurrences 1, 2, 3, 4, 5, 6, and these are these occurrences with these mutations on. So, then there are 8 occurrences, right. And it may be reasonable to assume that DNA A may also bind to these motifs with one of these mutations, right. It is not very unreasonable to think that way. And so we have this new computational problem, which is find the most frequent k-mers with mismatches in a string. Again, you are given a string text which represents the replication origin and integers k and d, right. So, where k is the length of the hidden message that you are looking at, length of the motif or pattern, and d is the number of mismatches that you allow. In this case, you are allowing one mismatch, right. And then all most frequent k-mers with up to d mismatches in text is what you are interested in. Again, I would like you to think about it as an exercise on what is the algorithm you would come up with and what will be the running time of the algorithm in a big O notation, right. So, how will it depend on the size of text k and d, right. So, that will be a good exercise for you. And again, this all most frequent k-mers is defined like before. So, you have a k-mer and you are looking for the presence of that k-mer or any other I mean either exactly or with at most d mismatches, and then you want to look at the counts of that, and you are seeing whether any other k-mer, the most frequent k-mer would be that one which, like, there are no other k-mers which occur more frequently than this one, okay, but there can be ties and there can be more than one such output distinct k-mer which occurs most frequently. So, if you apply this DNA boxes in equal a frequent 9 mers with one mismatch and reverse complements in putative replication origin of E. coli, right. So, then you get this particular 9 mer that occurs 4 times with mismatch of at most 1, right.